We've seen how the pyruvate dehydrogenase complex and the citric acid cycle generates the reduced electron carriers NADH and FADH2 in the mitochondria. And in this lecture, we're going to see how they pass their electrons to the electron transport chain in order to set up a proton gradient for ATP production. When we talk about how electrons flow, we really mean the passing of electrons from one molecule to another. And within the electron transport chain, it's going to be the change in redox potential that will determine what molecules are oxidized and which ones are reduced. There's also going to be a corresponding change in free energy that's going to be used to pump protons across the inner mitochondrial membrane and set up a proton gradient. This proton gradient is also known as the proton motive force because the difference in concentration of protons across the membrane is going to be the driving force for ATP synthesis. Peter Mitchell was the scientist that studied these processes extensively, and we'll look at his chemiosmotic hypothesis. This was established using an experimental approach, and we'll see how you can study oxidative phosphorylation in mitochondria in the lab. Check out the supplemental readings in the online Stryer textbook for another perspective on the key points for this lecture. The mitochondria really are the powerhouse of the cell. We saw that glycolysis generates 2 ATPs per glucose molecule, but this only accounts for 5% of the total ATP made in the cell. The breakdown of pyruvate, fats, and amino acids in the mitochondria can generate way more ATP. And this will all be due to the production of those reduced electron carriers, NADH and FADH2, that we saw in the last lecture. When they become oxidized, they pass their electrons to the electron transport chain, and it's going to set up a proton gradient that can be used by ATP synthase in order to phosphorylate ADP to ATP. I've alluded to the fact that metabolism can occur under aerobic conditions, and the reason why oxygen is so important for energy production is because oxygen is going to be the final electron acceptor in the electron transport chain. We'll also see that water is going to get made in oxidative phosphorylation both by the electron transport chain and ATP synthase. NADH and FADH2 are the reduced coenzymes that accept electrons in chemical reactions and then pass them through to the carriers in the electron transport chain. There will be a change in redox potential as these molecules are oxidized and reduced, and this is going to generate free energy that can be used to induce a conformational change in the membrane proteins. This will allow them to pump protons from the matrix into the intermembrane space. ATP synthase is then going to use this proton gradient to generate ATP from ADP in the matrix. I've made this brief animation to give you an idea of what's going on, and I will post it on Quirkus so that you can watch it a few times. NADH and FADH2 were made in the matrix by the enzymes of the citric acid cycle and the pyruvate dehydrogenase complex, and you'll see them donate their electrons to complex 1 and complex 2 of the electron transport chain. Complex 2 is actually succinate dehydrogenase that we saw in the last lecture, and it is the link between the citric acid cycle and the electron transport chain. So you'll see FADH2 coming in here and donating its electrons at complex 2, this membrane protein. What I want you to watch for is that you'll see the electrons moving from one carrier to another, and then protons will be moving across the membrane from the matrix to the intermembrane space. They'll then flow back through ATP synthase, and ATP will be made in the matrix. So complex 1, 3, and 4 are able to pump protons into the intermembrane space, and this creates a pool or high concentration that is the proton motive force. They're going to flow back down through ATP synthase, allowing for ATP to be made. Let's just watch it again. NADH donates its electrons. They travel through the chain all the way down to oxygen, which will finally produce a water molecule. FADH2 also donates its electrons, and we see more protons being pumped through the latter part of the chain. So electrons are going to be passed in the following manner. NADH is going to pass its So electrons are going to be passed in the following manner. NADH is going to pass its pair of electrons to complex 1, which is made up of numerous polypeptide chains and cofactors that can accept and donate electrons. It's also known as NADH-Q oxidoreductase, and the name gives you an idea of its function. There are going to be redox reactions where the electrons will eventually be passed to coenzyme Q. FADH2 was made by succinate dehydrogenase in the citric acid cycle, and this enzyme also goes by the name of complex 2 or succinate Q reductase, which will eventually pass its electrons on to coenzyme Q as well. Coenzyme Q are membrane-bound electron carriers that will then pass electrons onto complex 3, reducing cytochrome C, and then finally onto complex 4 or cytochrome C oxidase that will pass the electrons to oxygen, reducing it and picking up a proton in the matrix to make water. As these electrons are being passed from carrier to carrier, and there will be multiples of all of them, there will be a conformational change in complexes 1, 3, and 4, allowing for protons to be pumped across the inner mitochondrial membrane. These numbers along the side are redox potentials, and electrons are going to be passed from negative transfer potentials through to more positive ones. Let's look at what these numbers mean. The standard reduction potential is a measurement that tells you a molecule's tendency to be oxidized or reduced. You've probably learned about it in chemistry, where negative reduction potentials indicate that molecules tend to lose their electrons more readily or tend to be oxidized. A positive standard reduction potential indicates that the molecule likes to be in the reduced form. So when you have a pair of molecules, the electrons are going to flow from something that has a negative standard reduction potential through to a more positive reduction potential. When electrons are being passed from one molecule to another, there will be a change in redox potential, or a delta, and this can be used to calculate a change in free energy using this equation, where N is the number of electrons and F is going to be Faraday's constant. We measure the change in reduction potential in volts, so you can see that the standard free energy change will be in joules per mole. When we look at the standard reduction potentials, they're always written as these half-cell reactions, where, as the name suggests, molecules are being reduced. If you remember from chemistry, these were determined relative to a standard hydrogen electrode, and they'd be connected via a salt bridge, and we'd then use a voltmeter to measure the flow of electrons. If we look at the standard reduction potential for NAD+, it's accepting a pair of electrons and a proton in the form of a hydride to be reduced to NADH. The standard reduction potential is negative, indicating that NADH actually prefers to be oxidized and donating its electrons. When it comes to the final electron acceptor in the electron transport chain, oxygen, it'll be reduced to form a water molecule, and its standard reduction potential is positive, indicating that it prefers to be reduced. So if we look at the passing of electrons from NADH to oxygen in the electron transport chain, we would flip the first reaction and add it to the second, and this will yield the following net reaction. Do you remember how to calculate the change or delta for this net reaction? The change in redox potential as we pass electrons from NADH to 1/2 molecular oxygen is plus 1.14. Remember, to calculate this, it's always the acceptor minus the donor, or 0.82 plus 0.32. Now, I always get confused with these types of calculations, so I like to think of it graphically, where we're looking at the difference between a negative number and the positive number. There is a relationship between the change in redox potential and the free energy. So if you were to plug in plus 1.14 volts into this equation, you should see that this negative sign gives you a negative free energy change, so it is a favorable process. Passing electrons from NADH at the start of the chain through to oxygen, and this free energy change is what's going to drive the conformational change in the complexes to pump protons across their concentration gradient. So electrons are going to be passed from carrier to carrier in the chain, but do keep in mind that even though we do call it a chain, the electrons from NADH are going to go from complex 1 to 3 to 4, and those from FADH2 are going to go from complex 2 to 3 to 4. We can use the standard reduction potential to determine how electrons are passed through the different carriers, where good reducing agents will give up their electrons and have negative standard reduction potentials, passing their electrons to molecules that have more positive reduction potentials. The passing of electrons from negative to positive carriers will also result in a free energy change that's going to drive the conformational change seen in complex 1 and 3 and 4 to pump protons across the membrane. And it's the amount of free energy that will dictate whether or not 4 or 2 protons are pumped. These protons will then get used by ATP synthase to make ATP. I've just told you that electrons are passed from carrier to carrier, and this sets up a proton gradient that's used by ATP synthase. But how do we know that this is occurring in mitochondria in order to make the majority of ATP? Let's look at the experimental evidence and how you can demonstrate that the electron transport chain is linked to ATP synthesis, known in this process called oxidative phosphorylation. This was all determined by Peter Mitchell, a theoretical biochemist, 60 years ago when he proposed the chemiosmotic hypothesis that ATP synthesis occurs in the mitochondria due to an electrochemical gradient across the mitochondrial inner membrane. Chemiosmotic just refers to the difference in proton concentration across this membrane. When they were studying bioenergetics in the mitochondria, they couldn't find a phosphate donor with a high phosphoryl transfer potential, for instance phosphocreatine or PEP, that could be used by the electron transport chain to generate ATP. The electron donors, NADH and FADH2, were involved with establishing a proton gradient that could be used to generate ATP. Now, Mitchell also had a background in membrane biology, and he knew that ATP synthase was found in the membrane and that the integrity of the membrane was also important, allowing for this proton gradient to be set up. This membrane protein is also reversible, and he was able to set up experiments that studied mitochondria and demonstrate a link between electron transport and ATP synthesis. You can purify mitochondria and place them in an airtight beaker and measure the consumption of oxygen over time after you supply them with electron donors, such as NADH or succinate, which can be used to make FADH2. An oxygen electrode can measure the decrease in oxygen concentration, or the consumption of oxygen at complex 4, as we transfer electrons to 1/2 molecular oxygen and produce water. When he was studying this system, he noticed that the inclusion of ADP and inorganic phosphate was also important to allow this process to occur, but when you included detergents that disrupted the membrane, oxygen consumption was affected. We can also measure ATP production, and whenever the membranes were stressed or made leaky by these detergents, this was also decreased. There are also other chemicals that can be introduced to the system that help us understand the relationship between electron transport and ATP production. One of the tools that can be used to understand oxidative phosphorylation are inhibitors, or chemicals that prevent the passing of electrons or disrupt the proton gradient. Whenever you include one of these molecules, oxygen consumption or ATP production, or even both, could be affected. Inhibitors can block the passing of electrons through the different complexes. When either rotenone or amytal are included, electrons can no longer be passed through complex 1, and you won't see protons being pumped either, since the cofactors will be stuck in the reduced form. Antimycin A can block complex 3, while cyanide, azide, and carbon monoxide inhibit complex 4. So the effects of carbon monoxide poisoning are twofold. It inhibits the transport of oxygen by hemoglobin, so you won't have any of the final electron acceptor in the electron transport chain. But the complex itself might even not be able to pass electrons through to oxygen, reducing it to produce water. If we don't have the complexes working, they won't be able to accept electrons from other carriers upstream, and you won't be able to pump protons necessary to generate ATP through ATP synthase. Oligomycin is an inhibitor of ATP synthase, and it will prevent it from moving protons back into the matrix for use by the electron transport chain, as well as preventing the production of ATP. And finally, there are uncouplers that can disrupt the proton gradient, affecting ATP synthesis. Let's look at how these work. If you look at these structures, you might notice that they all have hydrophobic rings. This allows these chemicals to diffuse across the hydrophobic bilayer of the mitochondrial inner membrane. They also contain acidic groups that can be protonated or deprotonated, so they can pick up protons on one side of the membrane and move them across, and this will dissipate the proton gradient that's needed by ATP synthase. So we can use all of these different chemicals with an understanding of their function to understand this process called oxidative phosphorylation. So if you know that amytal blocks complex 1, how can you rescue electron transport and still ensure that ATP is being made? If amytal blocks complex 1, we can no longer accept electrons from NADH. FADH2 can still donate electrons to complex 2, which will pass them on to coenzyme Q and then through complex 3 and complex 4, which will pump a total of 6 protons. So we can rescue the electron transport chain by using a reduced downstream electron donor. ATP can still be made, it just won't be as efficient as when all the complexes are pumping protons. Now, if you were to inhibit complex 4, none of the complexes could pump protons since the chain's going to back up and all of the electron carriers will be in their reduced form. Let's take a look at how we can measure this experimentally. We can purify mitochondria and place them in an airtight beaker with some electron donors and measure the amount of oxygen consumed over time. This will demonstrate that the electron transport chain is working as oxygen is being used by complex 4 to produce water. If we add in ADP and inorganic phosphate, we would see an increase in the consumption rate of oxygen as ATP synthase is using the protons and returning them to the matrix, completing this cycle of complexes pumping them into the intermembrane space, and then ATP synthase bringing them back into the matrix. A plateau may occur if we run out of ADP, slowing down ATP production by ATP synthase. Let's walk through a few experiments. Let's set up the experiment and monitor both the electron transport chain by measuring the amount of oxygen consumed, as well as the activity of ATP synthase by measuring how much ATP is produced. At time 0, we'll start to see an increase in oxygen being consumed until we run out of electron donors. For the same sample of mitochondria, we will simultaneously see an increase in the amount of ATP being made, and there might be a bit of a lag as the proton gradient is being set up, but it would really come down to the sensitivity of your measurements. Let's run the experiment again, but say after 1 minute, we're going to add in some carbon monoxide and inhibit complex 4. We should see a decrease in the amount of oxygen being consumed as complex 4 is inhibited and the electron transport chain is going to slow down. If we were to measure the amount of ATP synthesized after the addition of carbon monoxide, we would also see a decrease in slope or the production of ATP as ATP synthase no longer has the proton gradient it needs to synthesize ATP, which was all set up by the electron transport chain. Now, what would happen if we inhibited ATP synthase? Let's run the experiment a third time, where we're going to add in oligomycin maybe after 2 minutes. Oligomycin is going to block the flow of protons through ATP synthase and slow down the production of ATP. If we're monitoring the electron transport chain, once oligomycin is added, the rate of oxygen consumption is also going to decrease, since ATP synthase is no longer bringing protons back into the matrix for use by the electron transport chain. We could, however, rescue the electron transport chain by introducing an uncoupler. When we add in an uncoupler like dinitrophenol, it's going to bring protons back into the matrix so that the electron transport chain can resume passing electrons and pumping protons. If we look at ATP synthesis, the addition of dinitrophenol will have no effect when we've got oligomycin inhibiting ATP synthase because it's essentially doing ATP synthase's job, bringing protons back into the matrix. And I do apologize for how messy this is, but I have posted a supplemental video for you to go over this again if you want. So we can use different inhibitors and molecules to understand the relationship between the electron transport chain and ATP synthesis. And you could even use them to figure out the order of how electrons are passed through the chain. Peter Mitchell also used detergents and saw how they inhibited ATP synthesis to come up with his chemiosmotic hypothesis. So in summary, we know that NADH and FADH2 pass their electrons through the electron transport chain, and this results in a change in redox potential that drives the pumping of protons across the inner mitochondrial membrane. Complexes 1, 3, and 4 will set up this proton motive force that's used by ATP synthase to make ATP in the mitochondrial matrix. We can use different inhibitors and uncouplers to see what effect they have on electron transport and ATP synthesis and understand how ATP is made in the oxidative phosphorylation. And finally, we've seen how Peter Mitchell's chemiosmotic theory outlines the relationship between the electron transport chain and ATP synthase, where you really do need this electrochemical gradient set up across the inner mitochondrial membrane in order for ATP to be made. Don't forget to do the brain break questions on Quercus after reviewing the material in this lecture.